Right, without further ado, we have with us tonight a very, very experienced presenter of the idea of intelligent design, and he's also been heavily involved in talks and conversations and arguments and everything else with anybody who would like to talk to him about intelligent design, and anybody who would like to talk to him about his own Christian religion. So, without further ado, 45 minutes, Jonathan convinces us about intelligent design. Jonathan McLatchie.
Well, thank you, Terry, for inviting me to speak here on intelligent design. I was connected with Terry through the Newcastle Skeptics in the pub a number of months ago when I gave a 10 or 15 minute presentation as part of an open mic night where I talked for 10 to 15 minutes about popular myths and misconceptions about the theory of intelligent design. And Terry very kindly and graciously invited me to come and present a more extended version to you guys. So here I am tonight. I'm a PhD student studying at the University of Newcastle, not far from here. And I study cell biology. I have 2 master's degrees, one in evolutionary biology, one in molecular biology. And my undergraduate degree was in forensic science. And so I'm fascinated with questions of science and religion, because I happen to be a Christian theist as well. And I'm a Christian apologist. And so I present reasons why I believe Christianity is in fact true. And I'm particularly interested in, as a scientist, the questions of how science can in fact support and provide evidence for belief in a creator. And so this evening I want to present some of the arguments, not exhaustively, we've only got 45 minutes, but some of the evidence for intelligent design, and hopefully clarify some misconceptions you might have about intelligent design, and respond to some of the common popular objections to it as well. So here's an outline for what we're going to be discussing. We'll introduce what intelligent design actually is, and just as importantly, what it's not. We'll argue that DNA contains information and explore some of the implications of that. We'll look at some of the molecular machines in the cell and how they present a formidable challenge to the neo-Darwinian evolutionary theory. We'll look at some of the common objections to design. And finally, we'll draw some conclusion. And of course, if I time things just right, hopefully we'll have no time for questions. Just joking. We'll have plenty of time for questions. So let's start by introducing intelligent design. So what's intelligent design? How do we define intelligent design? Anyone want to give me a definition of what they understand by it? Yeah.
God did it.
Okay. Anyone else?
Creation reflecting the creator.
Okay. Well, one popular definition, which we see promulgated by the popular media and press, is the following one. Intelligent design is the idea that living systems are too complex and improbable to be explained by the process of evolution, and therefore require invocation of a supernatural being to explain their existence, such as God. How many of you think this is a good definition of what intelligent design is? Quite a few of you. Okay. Anyone see any problems with this definition?
Yeah, I've got a problem with
Yep.
it. In terms of the fact that, okay, intelligent design, but it doesn't actually mean to say that someone has to design it. The design could exist on its own.
Okay. The key problem with this definition of design is the God of the gaps argument. It's an argument from ignorance, right? It's not framing the argument in terms of what we do know and making an inference based upon our knowledge. It's arguing from what we don't know and basing the argument upon our ignorance, what we don't know about explanations in science. So here's the better definition I want to propose, which does frame in terms of what we do know. Intelligent design is the idea that we can infer from patterns in nature that some features of living systems are best explained by an intelligent cause rather than a stochastic process involving chance and necessity. So immediately you can see here it's framed in terms of a positive explanation rather than simply being an argument from ignorance or God of the gaps, if you will. Now we want to know, to continue defining our terms, what is evolution? And of course, just as importantly, what is not. Here you can see Darwin's tree of life, which represents descent with modification from common ancestors. Now, the theory of evolution, if you read a modern evolutionary biology textbook, you'll understand that evolution is broken down into 6 main components. It can be defined as a change in allele frequency in a population over time. Non-controversial, an allele, by the way, is a gene variant. and it's non-controversial that allele frequencies in population change over time. We also can define it in terms of speciation, the idea that the species that are alive today are different from the species alive in the past, and species can arise by different mechanisms. There can be a geographical barrier, for example, which prevents species from intermingling with one another, and over time they become reproductively isolated and not able to interbreed anymore, and you have a new species. Evolution can refer to the proposition of descent with modification from common ancestors. The idea that all organisms alive, not just humans, but all extant species of organisms, were once are interconnected by virtue of common ancestors, and if you go back far enough, more than 3 billion years ago, you will find a single universal common progenitor of all life on Earth. Then there's the proposition of gradualism, the idea that life evolves by numerous slight successive modifications, to borrow language from Charles Darwin. It doesn't occur in leaps and bounds, it occurs by slight successive variations. Then there's the proposition of natural selection. Again, non-controversial. Natural selection happens, we know that. The idea that there's a selective force which preserves successful combinations of gene variants. The idea that, and by the way, commonly creationists will claim that evolution is entirely random. That's not the case. Natural selection is a non-random mechanism which selects for the successful gene variant combinations, the gene variants which allow the organism to survive in its environment. Then there's the idea of random genetic mutations, which provide the grist for the mill of natural selection. So, out of all these components of evolutionary theory, what is intelligent design challenging and what is it not challenging? Of course, it's not challenging this first one. We know that allele frequencies in the population change over time. We know that speciation happens, so again, intelligent design doesn't challenge this one. Common ancestry? No. In fact, intelligent design doesn't challenge common ancestry either. Contrary to popular misconceptions, intelligent design is compatible with common ancestry. In fact, many intelligent design proponents are advocates of common ancestry, and that there's good evidence for common ancestry. One of the key selling points of the neo-Darwinian synthesis is it purportedly explains the appearance of design without recourse to a designer. But simply providing evidence for common ancestry won't do the job. You could have common ancestry and still plenty of evidence for design in nature. Intelligent design isn't necessarily incompatible with gradualism, although it's consistent with non-gradualism as well, or subplationism. Intelligent design, of course, is compatible with natural selection. It doesn't challenge that natural selection happens. The key point that intelligent design contests is the sufficiency or adequacy of this one, random genetic mutations. The idea that life evolves and is driven by random genetic mutations which are selected by natural selection. Is random genetic mutation sufficient? Intelligent design argues no. How do we detect design in nature? Here's a cliff face. How many of you think this is a product of design? No one. Great. Of course not. It's a product of wind and erosion. What about this one? Anyone see product of design here? Of course, this is Mount Rushmore in South Dakota, where you can see presidents' faces carved into the rock. But you'll notice that this cliff face is no more improbable than this one. It's wholly improbable. Why do we know this is the product of design and this one is not? You foresee that this pattern conforms to an independently given pattern. You can see presidents' faces that we know were presidents of the United States. Here's another example to illustrate the point. This is Jodie Foster, who starred in the movie Contact, which is based on a novel by Carl Sagan, famous popular science writer, astronomer, cosmologist. And basically, the whole principle of this movie Contact was that the Search for Extraterrestrial Intelligence, or the SETI program, found radio signals which corresponded, coming from outer space, which corresponded to the first 20 prime numbers. And so they, of course, recognized that this wasn't the product of mere stochastic noise, but was in fact the product of an intelligent agency that was trying to communicate, an extraterrestrial civilization that was trying to communicate with our planet. Now, which of the following is designed? These are both random. This is a random assortment of 30 characters. But you'll notice this is equally improbable. 30 characters in the beginning was a word, which of course, if you're a Christian, you'll recognize this comes from John's Gospel, John chapter 1, verse 1. They're equally improbable, but we nonetheless recognize this one is a product design. This one could be explained just as well by random typing errors. Now, here's where I want to introduce to you the concept of specified complexity, which tries to explain to us how we determine and discern design in nature, how we distinguish design from non-design. This is a concept that was coined by Bill Dembski, who's a mathematician and philosopher. Specified complexity describes phenomena where, now these are the two criteria which are necessary for something to qualify specified complexity. One, the probability of a phenomenon being produced by chance alone exhausts the available probabilistic resources. And two, the phenomenon conforms to some independently specified pattern. So remember the cliff face example. You have not just an improbable cliff face, but you have a conformity to an independently given pattern, namely the faces of various US presidents. Or the example in the beginning was the word, again, not just a random signal, but you have the sequential arrangement of those characters forms a particular phrase which has meaning to us, and so conforms to some independently given pattern. And there you have specified complexity, and that in all of our experience, all of our uniform and repeated experience, only comes from intelligent agents. Just to illustrate this further, here's an ATM machine, which of course you're all familiar with. And you put in an ATM machine typically works, you put in your bank card, and you dial a 4-digit number on your PIN pad. And if you have 10 digits and you have a 4-digit PIN number, there's 10,000 ways of dialing in a 4-digit number, right? Typically, a bank will lock you out of your account after 3 trials, so that qualifies as the probabilistic resources that are at your disposal. If you have a random search, it is overwhelmingly more likely that you will fail to dial in your 4-digit PIN number, unless you know your 4-digit PIN number. And so that is what we mean by specified complexity. It's not merely improbable, but it conforms to an independently given pattern. Now, with this in mind, how do we reason about the past? If we look at past effects that maybe happened millions or billions of years ago, how do we explain them in terms of a causal explanation? Some of you might recognize this man. Anyone know who this is? This is a famous 19th century geologist by the name of Charles Lyell. And he wrote a book, which is very famous in the history of science, called Principles of Geology, an attempt to explain changes in the Earth's surface with reference to causes now in operation. And Charles Lyell insisted that if you want to explain effects in the remote past, one should let one's present experience of cause and effect guide one's search for the best explanation. This is called the principle of uniformitarianism, or the present is the key to the past. In other words, if you want to explain, for example, volcanic ash on an island, you favor the volcanic eruption hypothesis over the flood and earthquake hypothesis, because in every realm of cause and effect experience, volcanic ash comes from volcanic eruptions, not from floods, not from earthquakes, right? And so this is the principle of uniformitarianism, or as it's called nowadays, the abductive historical method, or the inference to the best explanation for multiple competing hypotheses. So with this in mind, let's look at some of what needs to be explained. So we have DNA. Life is chock full of this molecule that we call DNA, or deoxyribonucleic acid. And DNA contains information. Along the spine of the DNA molecule are chemical subunits which are represented by alphabetic characters A, T, C, and G, adenine, thymine, guanine, cytosine. And this sequential arrangement of those alphabetic characters conveys information. DNA does a lot of things. One of the things that a lot of the DNA does is encode for proteins. And the information in the DNA directs the construction of proteins. Here you can see a 2-part chemical factory known as a ribosome. And you can see the transfer RNAs, or the tRNAs, represented by these green structures here, which shuttle in these amino acid subunits. You can see methionine, arginine, serine. And the DNA has been taken in the nucleus and transcribed by an enzyme complex called RNA polymerase to messenger RNA. And the messenger RNA, unlike DNA, is only single-stranded, and it's shuffled outside the nucleus through the nuclear pore complex, and is taken to the ribosome, and the DNA, or the messenger RNA, is organized into 3-character words, or triplets, or called codons. And these codons each specify for an amino acid. There are 64 different codons, there are 20 different amino acids. And that determines the sequential arrangement of these amino acid subunits that form proteins. I'm going to show you this very short clip which shows you the mechanics of how this actually takes place. Okay, so here's the genetic code, which basically is the triplets of nucleotides and how they correspond to amino acids. And you can see there are stop codons, the 3 different stop codons. There's one start codon, which is the amino acid, or the codon triplet, which encodes for methionine. And so you can see there's 64 different codons there. There's 20 different amino acid subunits that form proteins. Now, the sequential arrangement of amino acids determines how a protein will fold into its 3-dimensional conformation. And so you can see here the primary structure of the protein, which is comprised of the amino acid chain, and the sequential arrangement of these amino acids will determine how a protein structure collapses into its 3-dimensional conformation. And so you can see here the secondary structure, which basically are called alpha helices and beta strands. And beta strands together form beta sheets. And then these together form the tertiary structure, also called domains of proteins. And these domains come together to form quaternary structures of proteins. So you have different levels of structure in proteins. Now, one question that we can ask, and both intelligent design and the evolutionary theory both make competing predictions about what we should expect to find here. So one question that we can ask, and we can discern this experimentally, is how big is a combinatorial space, first of all. There are 20 different amino acids used in cellular proteins, right? So that means for a monocyte length protein of 150 amino acids, there are 20 to 150 possible combinations. That's 20 times 20 times 20 times 20, all the way up to 150. Now this is the question that we can discern experimentally. So how prevalent are functional protein folds in combinatorial space? Now, intelligent design predicts that if these protein structures are really the product of design, we should expect the ratio of functional to non-functional sequences to be astronomically small. On the other hand, if the evolutionary story is correct, we should expect a relative abundance of functional protein structures within this vast array of combinatorial possibilities or ways of arranging amino acids. Now, this is a paper that was published in 2004 in the Journal of Molecular Biology. Its author, a friend of mine, Doug Axe, who at the time was completing his postdoc at the University of Cambridge, he published this paper in the Journal of Molecular Biology, and it's called Estimating the Prevalence of Protein Sequences Adopting Functional Enzyme Folds. This is not the only paper that bears on this question, it's an example, but other literature bears this out as well. And basically, he used a technique known as site-directed mutagenesis to randomly alter the protein structure of an enzyme complex, or a particular domain of an enzyme complex called beta-lactamase. And beta-lactamase is an enzyme which confers antibiotic resistance to certain bacteria. And here's the conclusion of his paper. He says, combined with the estimated prevalence of plausible hydropathic patterns for any fold and irrelevant folds for particular functions, this implies the overall prevalence of sequences performing a specific function by any domain-sized fold may be as low as 1 in 10 to the 77th power, adding to the body of evidence that functional folds require highly extraordinary sequences. And so it turns out that the ratio seems to be astronomically small. The ratio of stable and functional protein structures within that vast array of combinatorial possibilities, or that combinatorial space, if you will. And so that seems to be more consistent with what you'd expect given intelligent design than given the neo-Darwinian scenario. This presents the probabilistic resource problem. There are approximately 10 to 80 elementary particles in the observable universe. Since the Big Bang, roughly 10 to 16 seconds have elapsed. Now, the time it takes light to traverse the smallest distance is 10 to the minus 43 centimeters. And by multiplying those factors together, we can determine that the maximum number of events that could have taken place in the observable universe since its origin is 10 to the 139th power. So this is the absolute universal probability bound. Anything that exhausts this, you can rule out the chance hypothesis. This presents us with a needle in a haystack problem. Locating the functional sequences within the vast sea of combinatorial possibilities by a random search is like finding a tiny needle in an enormous haystack within a very limited time. And remember, natural selection is only effective once you've found these stable and functional protein structures. So natural selection won't solve the problem for you. Now, the problem extends deeper. when molecular machines require multiple cooperating proteins in order to perform their functions, the probabilities multiply exponentially. Here you can see DNA replication. And if you study DNA replication at either high school or university level, you'll know that it's made up of different proteins. You've got the DNA helicase, for example, which does the job of unwinding the double helical strands to prepare it for copying. You have the DNA polymerase, which does the job of copying, has a proofreading facility built in, or exonuclease activity as it's called. You also have the topoisomerase, which alleviates supercoiling. You have and you have various proteins involved. Ligase, which basically ligates broken DNA together, and so on. So there's a whole concert of proteins which have to be crafted in mutually specific ways to cooperate together in order to perform the job of a macromolecular machine. Now, I presented an argument that made use of a lot of intelligent design material in a debate recently at Monash University. And my agnostic philosopher opponent basically got up and said, well, there's still a chance that God doesn't exist, right? And I call this a Dumb and Dumber response. Now, if you've seen Dumb and Dumber, which is an American TV show, this is a scene here where this guy sees a pretty young lady and he says, you want my chances? And she says, not good. And she says, not good like 1 out of 100. And she says, not good like 1 out of a million. And his face falls, and then finally he gets really excited and he says, so you're telling me there's a chance? So I call this a Dumb and Dumber response to this argument. Now, as I said, the probabilities multiply exponentially when we're not dealing with just single proteins, but when we're dealing with macromolecular machines and indeed enzymatic pathways within the cell, biochemical pathways in the cell, which require multiple proteins to work together in very specific and coordinated ways. Charles Darwin, in his book, which he published in 1859, The Origin of Species, put forward a prediction that could potentially falsify his theory of evolution by natural selection. And he said, if it could be demonstrated that any complex organ existed which could not possibly have been formed by numerous successive slight modifications, my theory would absolutely break down. And it turns out that there are many such systems in the world of biology. Let me introduce you to another friend of mine, Michael Behe, who is based at Lehigh University. He's a professional biochemist. And in the late 90s, he published a book called Darwin's Black Box, where he sought to take up Darwin's challenge, and he coined a concept known as irreducible complexity. And by irreducible complexity, he says, I mean a single system composed of several well-matched interacting parts that contribute to the basic function, wherein the removal of any one of the parts causes the system to effectively cease functioning. And one of his favorite examples that he put forward in his book was the bacterial flagellum, which is an outboard rotary motor, which has many of the components that are present and found in a typical man-made motor, such as the propeller, a universal joint or a hook, the bearings and bushings, it has a rod and so on. And here you can see an electron micrograph that's taken of the bacterial flagellar motor. It basically propels bacteria like Escherichia coli or Salmonella typhimurium through liquid. And it's found, there's very diverse, flagella are very diverse, and you find a wide range of bacteria. Here you can see some animations of as it works. See, they typically rotate in bundles like this. Here's just an animation of the signal transduction circuit that's plugged into it, where it's getting signals from the environment, which they standardly rotate in an anti-clockwise fashion in response to chemical stimuli, such as glucose or repellents, such as poisons. They can shift gears and start spinning clockwise rather than anti-clockwise, which causes the bacterial flagellar bundle to break apart, and the bacterium flips in place or tumbles in place and can reorient itself in response to chemical stimuli or repellents. Here is it works. For some reason my videos aren't working today. Okay, let's get that one. So here's one of the things that's involved in bacterial flagellar assembly is rod penetration. So these things are actually remarkable in that they go far beyond what we've designed with man-made technology. They are they actually literally build themselves from the inside out. They build themselves within the cell, and they have an elaborate set of information and instructions to facilitate how it's put together inside the cell. One thing that needs to be done is rod penetration. In gram-negative bacteria, remember gram-negative bacteria have two membranes, an inner membrane and an outer membrane. And the the outer membrane in gram-negative bacteria serves as an impenetrable barrier that prevents rod penetration. And so there's there's 4 rod proteins: flag B, flag C, flag F, and flag G. And there's a rod capping protein, which is called flag J, which is bifunctional. One of its domains has binding affinity for the rod proteins, and the other one has hydrolyzing or muramidase activity, and basically punctures a hole through the outer membrane. It's necessary in gram-negative bacteria. Moreover, besides the 4 genes that encode rod subunit proteins, more than 10 genes are necessary for rod assembly. Then there's hook length control. Hook length across all bacterial species is constant at 55 nanometers. The hook acts as a universal joint. There's 2 important proteins involved, flag K and flu B, and the most popular model of how hook length is controlled is that flag K is secreted intermittently and acts as a molecular ruler that measures hook length. This is the way it works. The N-terminal domain of flag K goes through a channel and interacts with flag D, while the C-terminal domain forms a globular structure and stays in the cytoplasm. When the N-terminal domain reaches maximum length, it transmits a signal to the C-terminal domain to interact with flu B, which is the secretion gate. And then there's a conformational change that occurs to switch the substrate specificity. Then there's the filament structure. The filament, basically there's these flagellum monomers here, which are called fly C, have to be shuttled through a 2 nanometer diameter tube, which and the tube, the channel through which they're exported, is so narrow that they have to be literally unfolded for insertion into the channel and subsequently refolded at the distal growing end. And so there's this chaperone molecule called fly D, which is has been described as the most dynamic of all protein structures. It's composed of 5 leg-like extensions from this pentagonal disc, and basically because of an asymmetrical mismatch, it acts as a folding chamber for these flagellum proteins as they're shuttled through this 2 nanometer diameter channel. And fly D, by the way, is absolutely essential in flagella. If you look across the literature, that seems to be the case, that if you knock out fly D, then the flagellum monomers just get lost into the medium, so it's absolutely required, and in fact has no known homologs outside the flagellar system. Then there's a transcriptional hierarchy that determines how the flagellar apparatus is assembled. It makes no sense, for example, to start producing the propeller until you've already assembled the hook basal body apparatus. And so you can see that there's different ways in which this is done in different bacteria. This is the model system, which is Salmonella typhimurium or Escherichia coli, where you have 3 classes of operons. You have the class 1, class 2, and class 3 operons. An operon, by the way, is a sequence of genes which are transcribed together. And in the class 1 operon, you have just 2 genes, flu D and flu C. These together form what's called the transcriptional activation complex, or the enteric master regulator in the enterobacteria, like Salmonella. And these then, this transcription of these genes is regulated by a sigma factor called sigma 70, which binds to RNA polymerase and facilitates its expression. Flu D and flu C then combine to form this master regulator complex and activate the expression of the class 2 genes, which is 35 different genes over 8 different operons. These genes then express proteins necessary for the construction of the hook basal body apparatus, such as the different protein rings and so on, and the hook structure and the rod structure. And among those genes, there's flag M and fly A. Now, fly A codes another sigma factor called sigma 28, which then will bind to RNA polymerase and facilitate the expression of the class 3 genes. But it makes no sense to start producing this filament structure until you've already produced the hook basal body apparatus. And so there's another gene called flag M, which is expressed, and flag M basically sequesters or inhibits the sigma 28 sigma factor. And so it can no longer bind to the class 3 genes until it's time for it to do so. And so at the point where, as I mentioned, there's a substrate specificity switch inside the hook structure, the flag M actually gets exported out the needle structure, out the syringe, and then sigma 28 is then free, is activated, and is able to facilitate the expression of the class 3 genes, which includes the chemotaxis proteins, which are the signal transduction apparatus proteins, as well as the filament structure and so on. And so you've got a very well-regulated and coordinated and controlled mechanism for ensuring the timely expression of flagellar proteins. Now, there's good reason, I think, to take this to be an irreducible complex system. I'll come to an objection to that later. Now, common objections to design. what are some of the common objections to design? How might we respond? One of them is that intelligent design is a god of the gaps argument. But in fact, intelligent design, as I've explained, doesn't make an argument from what we don't know about explanations, but what we do know about the cause and effect structure of the world based upon our uniform and repeated experience of cause and effect. Intelligent design is based upon the standard historical abductive method of scientific reasoning. The present is the key to the past, where you reason from present experience to past causes. So intelligent design is based on standard principles of scientific reasoning. Another objection is that intelligent design isn't science. And there's a number of reasons that people say this. One is that intelligent design violates methodological naturalism. I want to suggest that it does not. Intelligent design does not violate methodological naturalism. In fact, it violates methodological materialism, which is something quite different. Intelligence is a natural phenomenon. It's not a material phenomenon. So I don't think it violates methodological naturalism necessarily. Another objection is intelligent design is not falsifiable. Whereas I would argue that intelligent design is falsifiable. You either show that, in fact, life doesn't contain specified complexity, or that specified complexity is best explained by natural material mechanisms, such as natural selection, genetic mutation, then intelligent design will be destroyed. In fact, most philosophers of science don't actually regard Karl Popper's criterion of demarcation, known as falsifiability, as a good criterion for distinguishing between science and non-science, because, for one thing, scientific theories and paradigms, generally speaking, are not falsified by a single anomalous observation, but it takes a lot of anomalous observations to really overturn a scientific theory. So falsifiability isn't really regarded anymore as a good criteria of demarcation. Then there's the objection, intelligent design isn't science because it doesn't make testable predictions. But intelligent design does make testable predictions. For example, it predicts that when we look at the world of the cell, we'll find specified complexity. That's a prediction that it makes. I've shown that it predicts things like irreducible complexity when we look at the cell. It predicts the relative ratio of functional to non-functional structures within that vast array of combinatorial space when we're dealing with proteins. Another objection is that intelligent design isn't science because it has no heuristic value. It's a sign stopper. Whereas I would argue that intelligent design actually encourages us to look for functions for, say, junk DNA, or maybe even things which are apparently suboptimally designed, which Darwinism doesn't. So actually it encourages and motivates further science rather than discouraging it. Another objection is that intelligent design isn't science because it invokes unobservable entities. But then other disciplines, such as particle physics, also invoke unobservable entities, so that won't do either. Another objection is that intelligent design isn't science because it doesn't address the modus operandi of design. But we still don't understand how consciousness interacts with the material world. I am a Cartesian dualist, so I would argue that the mind is not reducible to material constituents of the brain. But we don't understand how that happens, and nonetheless we know that agency is a real phenomenon. Moreover, if, say, an astronaut was to find a pile of machinery on the backside of the moon, or say a hieroglyphic inscription or something, he would be justified and quite warranted in inferring it was the product of design, even if he had no idea how it came to be there, or who designed it, or what have you. So that won't do either. Finally, intelligent design isn't science because a judge in Dover said so, which of course refers to the Kitzmiller v. Dover ruling in 2005, where a district judge in Pennsylvania ruled that intelligent design wasn't constitutional because it's religion and not science. But since when was it the place of a district judge to rule on matters pertinent to the philosophy of science? That's the job of philosophers of science, not district judges. So that won't do either. Another objection is a suboptimal design argument, and you can see here the famous recurrent laryngeal nerve, where you have the laryngeal nerve which comes down, loops around the aorta up to the larynx. And here you can see the famous inverse wiring of the vertebrate retina. But this is what I would call a converse God of the gaps fallacy, where you say, well, we can't explain why God might have designed it this way, therefore we'll plug in evolution as a placeholder to explain this gap in our knowledge. Actually, as proved to be the case on many occasions, many of these apparently suboptimal designs turn out to be optimally designed. And so the argument actually has to retreat with new advances in scientific discovery. For example, it's now understood why the vertebrate retina is wired the way it is. It's wired the correct way around in like octopus eyes, for example, or squid. Because these are cold-blooded animals and don't require the same blood flow to the retina. If it was wired the correct way around, if you will, in the vertebrate eye, then it would block the light, because the retinal cells require a large blood supply in vertebrates. Another objection is that incremental indispensability, or also known as the two-step Muller, or the Muller two-step model. Basically, it's the objection that this is an objection to irreducible complexity. The idea that, okay, so you could produce an irreducible complex structure by modifying or losing other components. So a structure that was originally only helpful becomes rendered essential by modifying or losing other components, right? The problem with this objection, it's best explained by giving an illustration, so let me give one. This is a mouse trap. Michael Behe used this as an analogy or an illustration for irreducible complexity. Imagine that you were to duplicate the spring of the mouse trap to produce a two-springed mouse trap. So a mouse trap, of course, has a spring, it has a base, it has a hammer, and so on, and it forms a complex system. Now, the rat trap it catches bigger rats, and so it has more force. It has two springs. So you've duplicated the spring. Now imagine that the first spring somehow becomes lost or mutated or modified. And so the second spring, now, although it was originally only helpful, is now essential. And so that's the way the argument tries to argue. The problem with this analogy, and I can give biological examples to illustrate if you prefer me to in the Q&A, is that you still require something performing the function of the spring. So I think so long as you define irreducible complexity, not in terms of structural components, but in terms of functions that need to be performed, I think this bypasses the objection completely. If you require the function of the spring, regardless of the identity of the spring, I don't think the objection actually holds water. Finally, we have an objection to irreducible complexity known as the co-option or acceptation objection. Now, Ken Miller popularized this. Ken Miller is a biologist at Brown University, cell biologist at Brown University. And he basically pointed out that actually, if you look at the flagellar system, you can see the type 3 export apparatus or the type 3 secretion system embedded. And actually, in the flagellar system, the type 3 export apparatus is what's secreting the flagellar proteins. Whereas in other species, or even within species with flagella, so for example, in Yersinia pestis, which is the causal agent of bubonic plague, which doesn't have flagella, the type 3 secretion system is used for exporting poisons or toxins into multicellular organisms. And so Ken Miller's objection is that you could produce a flagellar apparatus by adding to the type 3 secretion system, which you already have, and the type 3 export apparatus counts for about 10 or so of the proteins involved in the flagellar apparatus. What are some of the objections to this, however? There's actually some good phylogenetic evidence that the flagellum predates the type 3 secretion system. The type 3 secretion system actually seems to be an evolutionary degradation product. For one thing, the type 3 secretion system is taxonomically restricted to a small subset of gram-negative bacteria. Remember, those are the ones with two membranes. Whereas the flagella are much more widely distributed, which suggests that flagella is actually the predecessing system. Then selection for motility also precedes selection for virulence. So it makes the evolutionary sense that you have a selective pressure for it to swim before you have the selective pressure to inject poisons into multicellular organisms. Moreover, bacteria that have type 3 secretion systems, such as Yersinia, bubonic plague causal agent, possess genes for making flagella even if no flagella are assembled. Moreover, type 3 secretion genes are commonly found on large virulence plasmids, whereas flagellar genes are split onto 14 or so operons and are not found in plasmids. Moreover, when type 3 secretion genes are found in the chromosomes of bacteria, their GC content is typically lower than the GC content of the surrounding genome, and to biologists that suggests a foreign origin. And finally, the type 3 secretion system actually appears to be itself irreducible complex. A 2001 study, quote, examined the effect of loss of function mutations in each of the type 3 secretion associated genes encoded within that's the salmonella type 3 secretion system, on the assembly of the needle complex, finding that many of the type 3 secretion system components are required for the system's function. So in conclusion then, we've seen that intelligent design is based on standard principles of scientific reasoning about the past, or the historical deductive method, so it sits well within the bounds of science. We've seen that intelligent design challenges the concept of unguided chemical and biological evolution. We've seen that the design inference is based on cutting-edge advances in molecular biology, which reveals the complexity, elegance, and engineering prowess of cellular machines. Thank you. And here's my website here if anyone wants to check out my website, where basically I run an apologetics webinar every Saturday. I bring in speakers representing different viewpoints, and different Muslims, Christians, atheists, agnostics, what have you. and we have them present a short talk and then have a Q&A. And if anyone wants to contact me, there is my address there. Thank you.
Thanks very much, Jonathan. Okay.